And again, Jeanette really very beautifully summarized this. We have been using both linkage in families or association studies in populations to understand the genetic basis. And in the last 10 to 15 years, because of really, I guess, uh, technology, we've been able to do a lot of genome-wide association studies to understand the genetic basis of complex traits. And in fact, if you look at the GWAS catalog now, literally everything in the human genome is associated with a disease. These are all different disease categories. This is chromosome 1, this is chromosome 2, and every locus has some kind of an association with a disease now. But what we're really lacking is an understanding of these disease loci and their molecular or cellular mechanisms. This was a review article that was published a few years ago from Alice Chen Plotkin. And what it shows is at the end of 2016, there were almost 4,000 genome-wide association studies. And each of these studies actually discover sometimes hundreds of loci. So this bar, this blue bar, really represents tens of thousands of genome loci, genomic loci that are associated with a disease. But if you look at the number of loci that we have some kind of an understanding of for any complex disease, it's really lagging, right? I mean, there's a huge gap between what we understand at the biological and mechanistic level to the discovery, done with GWAS. So my lab is really trying to fill in this gap and focusing on the coronary artery disease loci. And we use an approach called systems genetics, but really the bottom line of systems genetics is actually pleitropy. So basically, if you have a genomic locus that is associated with two different phenotypes, then what you can do is you can just test the possibility that this genotypic effect on this phenotype is acting through another phenotype or not. And just the way I drew this, you can imagine that there's a mathematical way to use either linear regression with conditioning or Bayesian statistics to really figure out this causal relationship between the genotype and the phenotype. And I actually intentionally say causal relationship because the genotypes are obviously some things that we inherit from our parents and they don't change throughout our lifetime, right? Once you have the genotypic configuration, you have it for the rest of your life. So we can ask this question. And some very obviously, obvious examples is if you have a locus that is associated with coronary disease and it's also associated with LDL cholesterol, you know that that locus is acting through the LDL cholesterol to affect coronary disease, and similar for type 2 diabetes. But you can also imagine, instead of LDL cholesterol, we can do this with gene expression, right? We can ask if there's relationship exists between the genotype, gene expression, and coronary disease risk. We can, add layers to this. We can add protein levels and metabolite levels and so on and so forth. Basically, we can make this as complicated as possible and still try to find these relationships. So this is the most recent meta-analysis of about 550,000 people for coronary artery disease risk. And there are 163 genome-wide significant loci that are associated with increased risk for coronary artery disease. And if you go actually a bit lower, there are probably an additional 50 or so loci that are associated with the disease. And this is again from a review article that Dr. Erdman authored showing our understanding of these loci. As you can see, some of them are associated with lipid metabolism, and then we know that those loci act through the lipid metabolism. Some of them are associated with inflammation, so we know that they act through inflammation. But there are a lot of unknown loci that are here. And even some of those loci that what we consider that are known, transcriptional gene regulation is a very generic category, right? You don't know what are the cell types and what are the transcriptional targets and things of that sort. So there's still a lot of unknowns from the loci discovered in GWAS. It turns out that this was a paper that we published a while ago, but this held to be true. A lot of the variants actually affect transcriptional regulation. Very few of them uh, cause amino acid changes which might affect protein function. So many of these are actually in non-coding regions that are affecting transcription somehow. And the two most common ways that this happens is essentially either affecting an expression of a gene. So this is an example where you are, for example, heterozygous for a given locus, and in one locus, there's less gene expression from one of the alleles, and there's more gene expression from another allele. And the most simplistic, for example, way of thinking of it would be maybe an A here causes a less than perfect binding of a transcription factor in a promoter or enhancer, so then gene expression is less, right? So these are kind of regions, what we call EQTL regions, that stands for expression quantitative trait locus, meaning that it's a locus that affects expression, basically. Similarly, you can have another kind of transcription regulation through genetics, where this time this locus affects actually splicing. Here you can see that this A allele leads to the expression, higher expression of this gene isoform compared to the longer gene isoform. And this T locus leads to the expression of this, shorter isoform compared to the longer isoform. So these are what we call splicing quantitative trait loci. And again, the idea is simple. It's just a genetic variant that affects transcriptional splicing. So when we look at these studies that looked at, you know, transcription regulation of genes and look at how they colocalize with all these different traits, what you can see is for loci that affect coronary artery disease and myocardial infarction, many of the signal is coming from the artery. And for HDL, LDL, cholesterol, not surprisingly, it is from the liver and fat. Similarly for BMI and glucose, it's basically fat gene expression or genetic regulation of gene expression in these regions that are that there is an enrichment. So this result basically prompted us to think that, well, maybe some of the coronary artery disease variants, a subset of them that are not maybe associated with lipids, for example, actually affected this smooth muscle cell phenotypes, right? Because we know, I don't need to explain to this crowd the role of smooth muscle cells in lesion pathogenesis. So in order to test this hypothesis, we thought about a multi-layered approach, basically. So here we have the genetic variants, and then we wanted to identify how they are affecting transcription, how they are affecting protein abundance, and then put these things together, how they're affecting different, or how they're affecting the how these proteins and genes interact with each other, and then how that manifests itself as a cellular phenotype. And to answer this question, what we did was we isolated smooth muscle cells, aortic smooth muscle cells, from this ascending aortic region of heart transplant donors. So these are basically people who unfortunately passed away in a traffic accident, and their families donated their hearts to be transplanted into people who are suffering from heart failure. And when surgeons do these heart transplants, they have to cut pieces of the aorta to basically fit it into the recipient circulatory system. And what we do is we take these normally discarded tissue and actually isolate both endothelial cells and smooth muscle cells from these tissues. And for this experiment, what we did was we measured the amount of proliferation, the amount of migration, and the amount of calcification as a cellular phenotype. So these are obviously things that, properties of smooth muscle cells that play a role in atherosclerosis. And we did this under different conditions. Again, cytokines that have been shown to play a role in atherosclerosis and, regulate different kind of signaling pathways in smooth muscle cells. So these were our cellular characterizations. We also did molecular characterization. So we first of all genotyped these cells, and we have information from about 6 million genetic variants across the genome. We cultured the cells under two conditions, proliferative condition and a quiescent condition. So the quiescent condition kind of recapitulating a healthy artery, and a proliferative condition that's recapitulating a more, say, diseased artery where the smooth muscle cells are migrating and proliferating in the lesion. And we did RNA-seq for these cells. And we also looked at secreted protein abundance with LCMS because we know that smooth muscle cells lay down extracellular matrix proteins that affect fibrous cap stability and things of that sort. So I'm going to talk about some of these, not everything, because it's obviously way too much. But let me first talk about the cellular characterization. So again, I won't go into a lot of details, but in our detailed phenotyping, we had 7 proliferation phenotypes, 3 different migration phenotypes, and 2 different calcification phenotypes. And one of the first things that we observed was this. Let me also mention that our donor pool is actually a very diverse pool. A lot of GWAS is done on people of European ancestry, but we actually have a majority non-European ancestry population for our donors. And in the United States, a lot of the heart transplant donors are actually males, so that that's reflected in our donor pool as well. About 2/3, 3/4 of our donor pool is male, and we have 38 females. So one of the things that was really truly surprising to us was the amount of variation in these phenotypes across the donors. So what I'm showing you here are these different donors on the x-axis, and this is the amount of BRDU incorporation, which is, probably a little bit less than perfect metric for proliferation. And there is several hundred-fold difference between the cells from the donor that proliferates a lot compared to the cells from the donor that does not proliferate a lot. And I'm not going to show you for all 12 phenotypes that we measured, but this amount of variation was really very prominent in a lot of the donors. So that really kind of, that we scratched our heads because, honestly, we thought maybe we're doing something wrong to see this kind of variation. So we did an experiment where we grew cells from 4 different donors, and then we froze them in 2 different passages. And then we took them out and we did these experiments again, the migration experiment, proliferation experiment, and the calcification experiment. And we did it 3 times on 3 different days and weeks and all that kind of stuff to make sure that the variation that we're observing was not technical, that it was really donor to donor variation. And that's exactly what's observed. Actually, when you cluster these donors based on all these metrics that we measured, they cluster based on the donors. So the variation is coming from the fact that these individuals have different genetic backgrounds, and it's not so much that we're doing something wrong in our experiments. So there are some obviously interesting observations that I'm going to very briefly talk about these phenotypes. So, for example, PDGF is a well-known stimulant of smooth muscle cell proliferation, and that's exactly what we see. If you look at the amount of proliferation with respect to control conditions, when you treat the cells with PDGFBB, almost all of them, with the exception of just a few, really have increased proliferation. So this means there's 5-fold more proliferation compared to just control conditions where there are no cytokines, right? This is something expected, and we're obviously happy to see that our cells are responding to PDGF treatment. But there are also some interesting observations. For example, when we treat the cells with TGF-beta, some cells actually end up proliferating more than control conditions, and some cells end up proliferating less than control conditions. So TGF-beta is playing an inhibitory role for these cells and an activating role for these cells, right? So this is actually a very typical example of this gene by environment interaction concept, where the response to a stimulant is dependent on your genetic background, and which is what we observe here. But what's, I think, kind of interesting is that if you look at the literature, right, there are papers that says TGF-beta stimulates proliferation, and there are papers from actually the exact same group that says it inhibits smooth muscle cell proliferation. So we think that this is due to them using just different cells from different donors, and the genetic background is affecting these conclusions, basically. A question that I almost get is, so, you've done all these studies, can you look to see if a cell, for example, that migrates more, does it also proliferate more, right? Because in the atherosclerosis cartoons, right, that's always what we see. We see that the cells migrate and then they proliferate and then they calcify. So because we have cells from 150 people and we can look at this, correlation structure, it was kind of interesting that the correlations that you see are within a group. So you see high correlations when we measure different aspects of proliferation, but you don't see correlations with proliferation and migration or calcification. So cells that proliferate more doesn't mean that they're going to migrate more or they're going to calcify more. So maybe there are different, subsets of smooth muscle cells that are playing different roles, distinct roles in this atherosclerotic lesion progression process. Of course, we started with genomics, so the question is, are there genomic loci associated with these phenotypes? And it turns out that in fact there are. So despite us having only 150 donors, we were able to observe that there are 4 loci that are associated with different aspects of smooth muscle cell biology at the genome-wide significant level, which is denoted by this red line. These are, I think others have also shown these Manhattan plots where we measured, look at the association of individual SNPs with the phenotypes that we measure, and the y-axis shows the strength of that association, and these are the genomic locations of the SNPs. And there is no genomic inflation. This is always a concern when you have a small number of individuals, but we did not have this problem. And of course, if you look below this line of genome-wide significance, there are certainly many more loci that are associated with these phenotypes that we measured in smooth muscle cells. We were able to find some known biology through these association studies. So for example, one of the loci was a SNP on the ACTA2 gene that is associated with proliferation. And this is a pretty well-known, ACTA2 is obviously a smooth muscle cell marker, but not just that, but it also has been shown to play a role in smooth muscle cell proliferation. So this gives us the confidence that, okay, perhaps we're finding our findings are not completely irrelevant because we can find novel biology, and it gives us confidence for the rest of our findings, basically.
Mete, quick question. This is Claudio Villanueva.
Hi. Yep. Yep.
So ACTA2 is a marker for smooth muscle cells, right?
Yeah.
Do you think that sort of makes that connection with the observations of like PDGF being involved in TGF beta?
Well, yeah, this was actually for IL-1 beta stimulation, and indeed that is a possibility. What I didn't go into detail is there's apparently like a carg box in this first intron, and this SNP is really near that carg box, and that actually regulates ACTA2 expression. So it's probably a bit of a complicated mechanism as to why we're seeing this signal, and frankly, we haven't dug deep because I don't think we want to study ACTA2. But there's a possibility that, yeah, this is connecting the TGF beta, PDGF, and IL-1 beta signaling. The next thing we did was actually to annotate these loci using two different population-wide studies. One of them is the multi-ethnic study of atherosclerosis, which actually measured coronary artery calcification. And the other one is another study called AtheroExpress, which looked at carotid lesions and quantified various aspects of these lesions, like the amount of calcification, the amount of macrophage infiltration, the amount of smooth muscle cells in these lesions. And this was done in about 1,400 people, and MESA is a study of about 6,000 people. And one of the interesting things was that one of our loci that is associated with calcification was actually associated with the amount of calcification in lesions. And this is from the AtheroExpress study. And it was also associated with coronary artery calcium. And the direction of effect was very consistent among these three studies. So the allele that increased calcification in our study also increased calcified area and also increased coronary artery calcium. And when we look at this locus in detail, which is shown here, it is near this gene called SYNPO2, which also happens to be an EQTL, meaning that this SNP is also associated with its expression level. And it's been shown to, play a role in like smooth muscle cell biology. It hasn't been shown to play a role in calcification, but we think that this might be a novel finding in terms of vascular calcification, that this gene might be playing a role in vascular calcification. So of course, the main question is the coronary artery disease loci. So how about the coronary artery disease loci? So none of those genome-wide significant loci was associated with coronary artery disease loci, but there's a lot of signal, in this data set. So when we lowered the threshold of association, what we found was there were 79 of the 163 loci that had an association with a smooth muscle cell phenotype. And I'm just going to spend just a few minutes on this slide because it's a little bit complicated. But what I'm showing you on this x-axis are the SNPs, the most significant associated SNPs in each of the coronary artery disease loci. And these are the genes near those loci. And the color coding is such that if there is an increase with respect to the risk allele, then it's red color. And if there is a decrease with respect to the risk allele, it's in blue color, right? So first of all, you can see that there are some loci that are associated with only a certain phenotype. So these, for example, are a group of coronary artery disease loci that are associated with just calcification when we stimulate the cells with an osteogenic media. These, on the other hand, are associated only when we stimulate the cells with a media that contains increased inorganic phosphate. On the other hand, there are some other loci that are associated with proliferation and calcification, or proliferation and migration. So it can be pretty complex, right? There, some loci might have more than one role in the disease progression. The second thing that's also interesting is, not all the risk loci act in the same way, right? So if you think about this group of loci, right, this is an association with vascular smooth muscle cell proliferation. When we stimulate them with TGF-beta1, this group is actually is associated with increased proliferation when with the risk allele, and this group is associated with the decreased proliferation, right? So this is really all, I guess, speaking to the fact that a lot of these loci have very complex biology behind them, and it's not just that there's a one-way association that all the risk alleles increase migration. No, some of them increase migration, but some of them actually decrease migration. Is there a question?
Excuse me for interrupting. This is
Hi,
Klaus.
Klaus. Yeah. Yep.
How do you know which one is the risk allele in these in vitro studies? That was clear to me.
Oh, because we genotyped all the 150 individuals, so we know their genotypic configuration. And, for example, if you look at this, these are, for example, this is just for this one SNP, and we have 2 individuals with AA genotype and 117 individuals with GG genotype. And if we know from the GWAS that GG or G allele increases risk for coronary disease, then we can also find the directionality in our data set as well.
Thank you.
Yeah. So that was, and there's more, on this. Actually, we just published the paper. So I want to talk about, the next stuff that we're working on that, is not really published now, and this is getting ready for another publication. But there's a lot more detail on the paper about, how these loci might be acting to regulate, smooth muscle cells, and how some of these loci are affecting different aspects of smooth muscle cell biology. But remember, we also looked at molecular phenotypes, right, from these individuals. So we, again, as I just mentioned to Klaus, we have genotype information from about 6 million SNPs, and we did gene expression, in 2 different conditions from these individuals. We're still working on the protein abundance one, so I'm not going to talk about this. Hopefully, maybe in another actual meeting, I will present this stuff as well. But of course, the question is, when we do these studies, well, how representative is the in vitro culture to the in vivo condition? I mean, we're not really trying to say that we're capturing exactly what is happening in the vessel wall, right? But of course, we want to approximate as best as possible. So what we did is we clustered the expression level of thousands of genes that we have measured from these aortic smooth muscle cells with the 50 plus tissues that were measured in GTEx. And what you can see is that our smooth muscle cells actually cluster next to a muscle cluster that includes aorta and heart and things of that sort. So 2 things that, yes, our cells are probably representing some of the in vivo biology, but we can certainly find distinct things because there's obviously some differentiation from what is observed from these tissues that were obtained from cadavers, basically. If you focus further, because we did 2 different conditions, these are very distinctly separated in our smooth muscle cells, and there are thousands of genes that are actually different between these 2 conditions. So, and the typical culprits of like MYH11, calponin, etc., are all differentially expressed in the expected directions. But this tells us that we might be able to capture, again, different aspects of transcription regulation through genetic variation. And in fact, that's what we see. So this is again a little bit of a busy slide, but let me explain this. So we measured the expression level of nearly 19,000 genes. These include link RNAs and other non-coding RNAs and a lot of protein coding RNAs, obviously. And when we do that, we find that there are about 4,000 genes that have an association with genetic variants in proliferative conditions, and less so under quiescent conditions, about 3,000 genes. And we can do the splicing discovery on a subset of these genes, about 12,000 of these genes. And when we do that, we find that there are about 3,500 genes that are whose splicing is affected by genetic variants under proliferative conditions, and about 3,000 genes under quiescent conditions. And although there's some overlap, there's definitely distinct regulation, genetic regulation in different conditions. So this is showing the proliferative and quiescent conditions for both EQTLs and SQTLs. And there's, of course, we also looked at the possibility if a gene has both an SQTL and an EQTL, meaning both its expression and its splicing is affected by genetic variants. And what we find is that, you know, there is some overlap, but majority of the genes that have a splicing quantitative trait locus is distinct from the genes that have an EQTL, right? Both under quiescent and proliferative conditions. So this is a consistent finding under both conditions. Again, the question is, of course, what is happening at the coronary artery disease loci? So to answer this, we asked two distinct questions with two different methods. In the first method, we basically asked if the coronary artery disease loci and the variants, genetic variants in these loci share a causal mechanism with the expression and splice QTLs, meaning the same genetic variant affects either expression or splicing and coronary artery disease risk, right? It's the same genetic variant. In the second approach, this is a Mendelian randomization approach, we asked if the genetically regulated gene is causal for coronary artery disease. And you can do this essentially by separating your population into different genetic allelic configurations and ask if there is a causal relationship between the gene expression and coronary artery disease risk. Excuse me. And again, I apologize, you know, for this complicated graph. When you do these genome-wide studies, it's really hard to show them in, I guess, small figures. But what I'm showing you here at the top is the results from the Mendelian randomization study. And, you know, this is the red line in both of these cases is the genome-wide multiple testing corrected significance levels. And at the top, at the bottom, what I'm showing you is from the analysis of the causal variants, the shared causal variants. And these are the Bayesian probabilities that there's a shared causal variant. This is the statistic that shows that there's a causal relationship. And again, these are asking distinct questions, but of course, what we did is look at the overlap between these two results, basically. And we find that there are 15 genes whose expression is affected by coronary artery disease loci, and these are probable causal genes in these loci that are playing a role in smooth muscle cells to affect coronary artery disease association. What is really surprising was the amount of splice QTLs that we found to be regulated by the coronary artery disease loci. So 15 genes whose expression is affected, but 72 genes whose splicing is affected by the coronary artery disease loci. This is actually something that Jonathan Pritchard at Stanford had proposed, that majority of the GWAS loci are actually affecting gene splicing and not gene expression. And that's actually also what we observe in vascular smooth muscle cells as well. So we wanted to, of course, look at least one locus, to see if our genetic predictions are, whether they hold up or not, right? So we came up with a certain criteria to home into a few loci, and basically what we did was we started with the coronary artery disease loci. We overlapped those with the smooth muscle cell phenotypes. We overlapped them with EQTLs, and we also chose the loci that did not have an association with a known coronary disease risk factor like lipids. We wanted to make sure that that we were not confounded by regulation and for lipid levels. And we focused on one locus, Mia3, and I'll explain why. Because what we observed was the risk allele in our study was associated with lower proliferation of vascular smooth muscle cells. And this allele, the same allele, was also associated with lower expression level of Mia3, right? So lower proliferation, lower expression of Mia3. But we could also observe this exact same relationship between gene expression and this SNP in an independent data set of human aortic tissue. And in this independent data set, again, the risk allele was associated with lower levels of Mia3. And all of these basically tell us that then there has to be a positive correlation between the expression level of Mia3 and the proliferation in smooth muscle cells, and that's exactly what we observe here. Mia3 has been studied in cancer cells. It is a protein that localizes to the ER exit site, and it has been shown to actually affect collagen secretion. So, it kind of was an attractive candidate, knowing the role that collagen plays, of course, in plaque stability. So we wanted to create conditions that were similar to the risk condition. So we took cells from 3 different donors and repressed Mia3 expression with 2 distinct shRNAs, which essentially recapitulated our genetic findings, because the repressed gene expression led to lower level of proliferation in smooth muscle cells. We then collaborated with Gary Owens, because he has this mouse model where the lesions of these Oct4 deleted mice in the smooth muscle cells have these unstable lesion characteristics. So we stained the lesions with Mia3 and several other markers. I'm not showing everything here, but certainly ACTA2. And then we quantified the amount of Mia3 positive cells that are, smooth muscle cells in just the fibrous cap. And what we observed was that in the lesions with these unstable lesion characteristics, there were fewer cells that were positive for Mia3, fewer smooth muscle cells that were positive for Mia3. Again, kind of going in this direction of lower Mia3 levels is associated with, increased risk for coronary disease, increased risk for maybe an unstable fibrous cap. And finally, we also looked at human coronary artery lesions. For this, we collaborated with Renu Virmani, and she and her group looked at basically lesions that have both thick caps and thin caps, and stain them for multiple various measures of lesion stability, and also Mia3 and smooth muscle cell alpha actin and macrophage markers. And what we observe here is that the thin fibrous caps in the coronary arteries had actually fewer Mia3 positive smooth muscle cells. Once again, kind of corroborating our genetic findings. So just to, I guess, recap, this I think is a pretty good resource, and I'm obviously happy to share our data if those of you who are interested in vascular smooth muscle cell biology, because we did an extensive characterization for both gene expression and these phenotypes, and both for gene expression and phenotypes we observe a large amount of variation that lead to several genome-wide significant loci, and some of them are actually definitely acting in the coronary artery disease loci. And specifically for this MIA3, we think that the risk allele is associated with lower expression, and that lower expression is perhaps affecting some fibrous cap characteristic. So let me acknowledge all of our collaborators. This was a large study. A lot of people shared data with us, and we're very grateful to them. And I also really want to acknowledge Dr. Redwan Aharau, who is finishing up his postdoc here. He is very happy after the completion of all of his cell culture experiments. He really did nothing but cell culture and cellular phenotyping for almost 2 years. A very dedicated, hardworking, and a patient postdoc. And he was aided by an army of undergrads for this work. And I want to acknowledge them, and also other folks in the lab whose work I didn't really present, but they're working in this area of functional genomics. So thank you so much. And I'm really happy to take any questions from you.
Okay, great. Thank you, Mete, for this great talk. Now we can take questions. Please use the raise your hand function at the bottom of your screen.
I can't raise my hand. Can I go first? Because I can control the mute button. Sorry, I can't raise my hand because I'm a co-host. Mete, that was spectacular and amazing demonstration of, the tour de force of functional genomics and trying to understand just the many ways that these things are contributing to disease. So I guess maybe I missed it. It's very possible I missed it, but for MIA3, again, obviously the identification of it and its functional outcome, but was it one of the ones with a splice, like a splice variant? And did you check if it's the different splicing variants that are the sort of causal, so to speak?
Really good question. It has actually multiple different splice variants, but this was an EQTL. Okay. And we think, there's only one abundant isoform in smooth muscle cells. So we think that this is actually affecting expression level and not splicing level. Not the splicing of it. Okay. We have other, splice QTLs that we are interested in, but, they're also a little bit harder to study.
Right. Thank you. Okay. I'll ask a quick question, Mete. You show that your population has 1-to-3 ratio of female to male and also has different ethnicities. So your findings, for example, with proliferation or calcification phenotype, are they more pronounced in one sex or ethnicity? Did you do sub-analysis?
We are, literally this week we started doing it. Yeah, Redwan, and we have a collaborator, Hester, in the Netherlands and Utrecht, and she wants us to look at that. But basically what we're gonna do is we're gonna do a gene or SNP by sex association study. The problem is we couldn't do it separately because the power is so different. There's 113 males and 38 females. So the allele, the frequency is very different in the two populations. But we're going to use the entire population to look at if certain genetic variants affect both gene expression and phenotypes in different sexes in different directions or in different effect sizes, like more in one sex or less in another sex. But hopefully I'll present that data at some other point.
Okay, great. We have, yeah, go for it.
Hey, Mete, good to see you again.
How are you? Good seeing you.
Good, how are you? Yeah, we just discussed last week. Yes. Again, excellent work. And
thank you.
I have a follow-up question about the MIA3 as well. So it seems like this MIA3 also may play a role in regulating the plaque stability. Do you know what's the function? Maybe I missed that part. Does it regulate the muscle cell like migration, proliferation, or have anybody look at it?
Yeah, we haven't. So based on literature, we think it's affecting collagen secretion. Okay, and our evidence for that is actually just correlations with the expression level of collagen genes, which is not a perfect way of testing this. So we haven't tested yet, but it seems, and based on its role in this ER exit sites and the COP2 vesicles and stuff, that seems to be the mechanism that we still need to test.
Okay, thanks.
Yeah.
Ada, go for it.
Thank you. That was very interesting. Thank you. I was wondering the sequencing of the DNA, on which cells do you do it? And do you see any somatic mutations in the smooth muscle cells? And can that infer some of the phenotypes that you're seeing?
Yeah, that's an excellent question. So the genotyping we did was from these cells, from these smooth muscle cells. So when we culture them, we take a portion to do DNA isolation and do the genotyping that way. So since we don't have blood from the donors, we're not in a position to compare to the blood genotypes and see if there has been a somatic mutation. But if there has been a somatic mutation, right, we're capturing that basically because we're just looking at the genotype of these cells. But whether cell culture caused some somatic mutation, I think that's an excellent point. That really is an excellent point.
Right.
Okay.
Can I ask a question?
Sure, go for it.
Hey, very good talk. Congrats on the secretion research paper. I learned a lot.
Thank you.
Good job. So I know lots of people in small muscle field focus on the migration proliferation. Did you pay attention to inflammation as well?
Yeah, not really. For two reasons. I mean, one is we're hoping to do that with the gene expression because when we actually, Noah, for example, who's a grad in the lab, created networks from this gene expression data, and there's definitely an inflammatory signal there. And the other one is actually a practical reason. Really, when you're doing this many cell cultures and phenotyping, you just have to be cognizant of how many things you can do at one time, really. And we're I guess, limited by that a little bit because they have to be done in a relatively high throughput manner. I mean, Red One doesn't want to be in the lab for 10 years doing these experiments. So we did not look at an inflammatory component, to be honest.
The reason I ask you because you used the interleukin-1 treatment, right? You must have some reason behind this.
Oh, the reason for those cytokine treatments were for sure that they have obviously been shown to affect various smooth muscle cell signaling pathways in vascular smooth in the progression of atherosclerosis, but we are not necessarily looking at some kind of an inflammatory, a monocyte interaction or something like that. We're just looking at the IL-1 beta effect on proliferation, for example.
All right, second question. So did you pay attention to those non-coding regions that's affecting some link RNA or non-coding, like messaging especially?
Yeah, so if I think maybe I can show here, some of the coronary artery disease loci are actually definitely affecting link RNAs and link RNA splicing. So if you look at this list, there are some link RNAs here. If you look at this expression list, there are some link RNAs here as well. I think maybe not in this condition, but I think in the splicing condition. So yes, indeed, there are link RNAs. They are, as you know, they're relatively lowly expressed. So we did 100 million reads per donor. So that's quite deep sequencing. But even then, we measured expression of a few hundred link RNAs.
All right, thank you. Thank you.
Okay, we have two more questions. Andy, go for it.
Hey, Mette, how are you?
Great seeing you. I haven't seen you in years. I love
so my
this
question
metabolism series.
So that extreme donors with extreme phenotypes do they have extreme genetics? Is it genetically driven that they have an extreme phenotype, or environmentally?
Yeah, I mean, we certainly think it is genetics, but we haven't done any kind of exome sequencing to see if there has been some kind of a mutation or something like that that led to that extreme phenotyping. And even if we did, I guess we would still have to have multiple donors with extreme phenotypes to be sure that, you know, you can find evidence for the same genetic variation in multiple donors, right? Because if we whole genome sequence these people, you're going to find a ton of what we call singletons, right? The private mutations. Whether that is the cause of that extreme mutation or not, we don't know. Yeah.
Do they have an overrepresentation of risk loci from the loci that you've identified?
Oh, that's a great question. That was in the paper. So when we did that with a polygenic risk score and genetic risk score, and we find an association with migration. So I can't remember now. I think it was actually inverse correlation. So people who had more risk alleles had lower migration than people with fewer risk alleles. But we did not find an association with other phenotypes, which was kind of interesting. Holger has a question.
Mete, beautiful work, and congrats to you and Ridwan. That's really awesome. I have a question regarding the MIA3. Does it go both ways? Did you see actually patients with gain of function mutations? They have an overexpression of MIA3, and do you see that these smooth muscles produce more collagen? Are they more protected?
Yeah. So I mean, remember that in my mind, gain of function is an amino acid change, but obviously we're looking at a change in the sequence that affects transcription. So the people with the risk alleles, obviously there is a variation, right, in their expression level. So this person carries the risk allele and this person carries the risk allele, or vice versa, and they still have very high levels or different distinct levels of Mia3. Now, we didn't compare, like, say, within group, like, say, these three to these three. But I think that's a really good point. I think we can definitely do that and see what difference that has on it. But we always consider this variation is because, Mia3 is regulated by so many different things. It's not just this one SNP. So those are causing this variation.
Mm-hmm. Oh, yeah,
that's
great.
a good point.
Yeah, thank you, Mete.
Okay, I guess that's the end of the questions. We thank Mete again for this great talk, and all of you for stimulating questions. And we hope to see all of you next week, same time.
Thank you. Thank you, everyone.
Bye, everyone. Thanks, Mete.
Bye.
See y'all
Bye.
next
Thank
week.
you.
Thank you. All right. And are you guys going to continue? Do you want me to make you host? Okay. All right. Bye, everyone.